We'll be starting in another 2 minutes. Yeah. So let's other join. So good morning, everybody. We'll be starting in another 2 minutes.
Good morning, sir.
Good morning. Good morning. Yeah. We'll be starting in another 2 minutes. Yeah. So let's other join. So okay. So good morning. So once again, and let's start today. Today we are supposed to learn two practicals, which we are supposed to do in the lab, but due to lockdown, we were unable to do that. Okay, so good morning. Can anybody suggest means how I can minimize the two means like I'm speaking in Zoom, so the sound is coming twice in YouTube also. Can anybody suggest how to do that? Minimize the sound or Okay, good morning, everyone. Once again. So today we are going to start the lab uh, course for life science for engineers, which we were supposed to do. So the first practical in this regard is my use of microscope, that is microscopy. A microscope is a laboratory instrument used to examine objects that are too small to be seen by the naked eye. So here are the parts of an ideal microscope, which is seen here. So microscope consists of an eyepiece, which is shown here, and then it has a head structure which supports the eyepiece. Then this portion is called Aperture adjustment. Through this, one can adjust the eyepiece according to his or her convenience. Then there is portion round circle portion called nose piece, which is attached to objective lens, and these objective lens are of various sizes, starting from like 10x, 40x to 80x, 100x, and the portion which supports the eyepiece along with the other parts of the microscope is called arm. And here there is a stage where the sample with which we want to see the object is placed here, and this is called stage clip. It is movable. One can pull it and then attach the slide or the object which needs to be visualized by the microscope. Then there is also an aperture. Aperture is used to control the light intensity so that the amount of light which is entering the object can be controlled. And then this is the mechanical stage where the object is placed. Then there is like below the stage a diaphragm is there. Diaphragm is used to control the light intensity which is coming out of the light source, so that it can light intensity as well as the size. Then condenser, which is present below the diaphragm. What it does is that it controls all the light wavelengths so that the light can come out in a single ray and it can fall on the object, and then it can be visualized through the objective lens. Then there is illuminator, through which the light source comes out. And then there are two adjustable things which can adjust the light source as well as, so that is, one is called coarse adjustment. Through this, one can change the adjustment very fast, and this is one fine adjustment through which the adjustment can be done in a small scale, and it is also called fine adjustment. And through this stage control, one can lower or raise the stage. And then this is the base of the microscope. That there are two switches. One is for light switch and the other is for brightness. So through this brightness as well as through the diaphragm, one can control the intensity of the light. So am I audible to everybody?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Sir.
Am I audible? Oh, okay.
Yes, sir.
Am I audible now?
Yes, sir.
Am I audible now?
Sir. Audible, sir.
Okay, thanks. Yeah. So now we said that there are various parts in a microscope. The parts starting from eyepiece, objective lens, then diaphragm, light source, and through this we can actually connect it to electricity, and then we can either switch on the microscope or switch off the microscope. So the two parts which were very important in microscope, one is diaphragm and the one is condenser, which we just now saw. So diaphragm is present here just below the stage and above the light source, and condenser is present below the diaphragm and above the light source or the illuminator. Let's see what are the function of diaphragm or the condenser. So diaphragm, many microscopes have a rotating disc under the stage. This is this portion. This is rotating disc. It can be rotated. So this is diaphragm, and what it does is that it controls the intensity and the size of the light that is projected into the slide. That means the area which the light is covering on the object can be controlled, and the intensity, bright light, dim light, which is falling on the object to be seen under the microscope can be controlled through the diaphragm. So the function of diaphragm is to control the light and intensity and the size of the light which is falling on the object. Then there is condenser. So condenser, what it does is that it is present below the diaphragm, and it serves to gather the light source and concentrate them into a cone of light that illuminates the specimen. So what it is does is that so light source is coming out of the illuminator. So condenser will condense the light source into a single stream, single beam of light which can fall on the object, and it will help in the better visualization of the object under the microscope. So now today we are going to see how to use a microscope and what are the different parts of a microscope. So let's see through this video. If I can play that video, hopefully it will be clearer. So let's hope. Just wait, I am playing. Just selecting. Through this video, one can see how to use a microscope specifically.
Video audible is not audible, sir.
So I have shared a link on the chat. So please use that link. And oh, it's a private message. Okay, sorry. Yeah, wait. Yeah, I can. Everyone in the meeting. I have shared a link in the chat box. So please use that link to watch the video that will say you how to use the microscope and the components of the microscope. Once everybody is done watching the video, we will start with the another portion in the lecture. So please, I suggest everyone to use the link to watch the video, because since there is some problem in telecasting the video, somebody cannot listen the sound. So I have shared the link. So please use that link to watch the video. So once that video is over, we will. Can anybody tell that whether they are able to use the link to watch the video or not? Please.
Yes, sir.
Yeah. So please, yeah. So watch it and then we can proceed with the other thing. Yeah. So let me start from the beginning. Yeah.
Correct. Yeah. Probably just my fine. I think in all three of the objectives.
So hopefully everybody saw the video, and from the video it was very clear how to use the microscope. The video was really good because it shows you clearly the minimum basic parts of a microscope and how a microscope can be used to visualize an object. Furthermore, just for like remembering it once again, so you can see that the microscope contains an eyepiece, then objective, and eyepiece and objective are the two crucial factors for seeing an object and magnifying it so that you can see it through your eye. So for magnification, two things are important, as was said in the video, like eyepiece and objective. Okay, then the other important part is diaphragm, through that which you can control the light intensity. The next important part which was shown is this, the coarse adjustment and knob and the fine adjustment knob. Through this you can move the stage up and down, and then you can adjust so that you can visualize the object better. Further, the same thing can also be done by this portion, which is stage control. If you will move this up and down, and this also can be used to move sideways. So these were the parts of the microscope, and then you saw in the video how to use the microscope. Let's see once more. Now we will see how we can use the microscope to visualize something, or how we can use the microscope to study mitosis. Mitosis we have studied in our theory classes, and we have seen that mitosis is a process where a single cell divides into two identical daughter cells. So this is a very simple process, and it happens in all prokaryote as well as eukaryote. By this process, a single cell gives rise to two identical daughter cells. So let's see in brief what all are there in mitosis. So mitosis consists of many stages. So it starts with the stage called interphase, then prophase, metaphase, anaphase, telophase, and cytokinesis. We have studied these steps in our theory classes, but for time being, just see what all in brief is there present in each and every stages. Like in the first phase is interphase. In interphase, the cell prepares itself for undergoing mitosis. So if you see an interphase cell, you can see cell membrane, centrosome, nucleolus, cytoplasm, chromosomes, and nuclear membrane. But once the cell enters into the prophase, you can clearly see the nuclear membrane. If you see this line, the nuclear membrane which was there, it starts to disintegrate in the prophase, and the chromosomes are visualized properly. That means chromosomes are more condensed, and you can see the chromosomes properly, like sister chromatids, okay, which was not visible in the interphase nucleus. Furthermore, you also starts to see spindle fiber, which was not visible in the interphase. Then once the cell passes through the prophase, the cell enters into the metaphase, and the major characteristics of metaphase staged cells are the chromosomes are aligned on the center or the metaphasic plate of the spindle fiber. So you can see clearly, so the spindle fibers, these are the spindle fibers, and the chromosomes have aligned centrally on the metaphasic plate. Then after metaphase, the stage which is there is called anaphase, where actually the chromosome starts to move in opposite direction towards opposite poles. Okay, so if you can see here the green or the blue chromosomes, which are marked for better visualization, you can see these chromosomes are moving towards one pole, and here these chromosomes are moving towards this pole. So anaphase means the movement of the chromosome towards the opposite poles, and metaphase means the chromosomes are aligned on the center, and prophase means the chromosomes are condensed so that they can be visualized. The nuclear membrane disintegrates and the spindle fibers starts to form. And what happens in telophase is that telophase, the chromosome separation has been completed. So you can see the chromosome have reached, the spindle fibers disintegrates, and the cell starts to forward, the cell starts to divide. So the then after telophase, complete two identical daughter cells are formed from a single cell, and you can see nuclear membrane again comes back, okay, then again centrosome comes back. So if you want to recapitulate whatever we have told just now, is that there are basically four different stages of mitosis. First is prophase, metaphase, anaphase, and telophase. So prophase is characterized by condensation of chromosome, where you can clearly see the chromosomes, okay. The nuclear membrane disintegrates here. Then in metaphase, the chromosomes are aligned on the metaphasic plate. Then in anaphase, the chromosome starts to move towards opposite poles. In telophase, the chromosome separation has been completed. Okay, so these are some of the salient features of each stage of mitosis. So when one sees under a microscope, the mitosis looks like this. In prophase, you can see condensation of chromosome, and the chromosomes are clearly visible. Okay, this is the special characteristics of prophase cell. And metaphase, you can see all the chromosomes have been aligned on the metaphasic plate at the center. Okay, so this is the special characteristics of metaphase. Then in anaphase, the chromosomes are moving towards the opposite pole. In telophase, the separation has been completed and the separation of the cell into two identical cells have started. If you see carefully, here there is a furrow kind of structure and there are two cells clearly. There is two nucleus. Okay, so these are the salient features of mitosis. Now we will see a video which I will share the link also in the chat box. So please use that video to see how one can observe mitosis by using the root tip of an onion. Okay, so this is the animation video. So it will help you to better understand. So let's go to the video. I will share the link of the video in the chat box so that you people can better visualize. So through this video, we will see how to use the microscope for studying mitosis. Then we will proceed further. So let's see what this video and how to do that. Yeah. So just now you all saw how to observe mitosis using onion root tip. But now there was a small problem in the video which was shown to you. The problem was that you saw what all the requirements for starting to observe mitosis in under the microscope. So like you needed an onion root. So what you did in the beginning is that you place the onion into water for 3 to 6 days so that the root tip starts to grow. You were doing this because this helps new roots to form, and it is very easy to detect mitosis when the root tips are young. That means they are recently grown. Okay, so that's what you are using. So you saw there were some chemicals, like one was like acetic acid and ethanol mixture. So that is basically used to fix the root tip. Okay, it is used to fix the root stages of the cells where they are. That means if suppose some cells will be there in mitosis or some cells will be there in prophase, they will be fixed at those stages only. They will not be dividing anymore once you have put that acetic acid and alcohol mixture. The second important reagent which you saw was acetocarmine. Acetocarmine is an stain which is specifically used to stain the chromosomes, which you are seeing right here. Like if you see blue color thing, so that is specifically what it does is that it stains the chromosome. Now, so what you saw is that in the video, a root tip was taken. It was placed on a cover slip. The cover slip uh, slide, first slide, the rectangular structure, glass, it is called slide. You were putting a small tiny root tip on it. And then first thing you were doing is that you were washing it with water. After washing it with water, then what you were doing is that you were fixing it with acetic acid and alcohol mixture. Then after that, what you do is that you were putting zero point 0.1 normal hydrochloric acid, and after that you were putting the acetocarmine stain. Once that is done, you were putting it above a Bunsen burner flame so that to dry the liquids, and after that you were seeing under the microscope to visualize the different stages of mitosis. In the video there was a small problem, is that what the problem was is that first they were showing the staining with acetocarmine, and then at the end stage they were showing it water. So that was the problem. Water washing will be done at the beginning, and the staining with hydrochloric acid 0.1 normal and then acetocarmine will be done at the later stages, and you will observe under the microscope only after you have put the stain acetocarmine. And hydrochloric acid is used because it helps in the better staining of the chromosomes once acetocarmine is added. So let's see one more video which will make the process understanding more better. Okay, so this video is also for studying the mitosis in the root tip. So let's see. Hmm. Hmm. So we saw how we can observe mitosis using a root tube under the microscope. So by now, everyone should be clear. So what are the different stages of mitosis? Like prophase, metaphase, anaphase, and telophase. Prophase, the chromosome condensation begins, so you can visualize chromosome more easily. Then metaphase. Metaphase, the chromosomes are aligned on the center metaphasic plate, and then anaphase, the chromosome starts to move away from each other under two opposite poles, and then telophase, the chromosome separation stops and chromosomes are at two poles apart, and cell also starts to divide. So let's see it once more. Okay, mitosis consists of prophase, metaphase, anaphase, and telophase. There is one more stage called interphase, which is actually before prophase, and where what does the cell do is that the cell prepare itself for cell division. Then prophase. So prophase is where chromosome condensation happens, and one can visualize chromosomes properly during that stage. That is called prophase. In metaphase, chromosomes aligned at the metaphasic plate, okay, of spindle fibers, or at the center of the spindle fiber in layman language. The third step is the anaphase, where the chromosomes actually starts to move away from each other in the opposite poles. Then telophase, the chromosome separation is completed and the cell is about to divide. And you can see that after telophase, there is a stage called cytokinesis. Cytokinesis is the division of the cytoplasm, as a result of which two cells will be formed. Okay, so mitosis, which is a division of a cell into two identical cells, is completed now. So till now you saw what are the different parts of a microscope and how microscope is used in the laboratory to visualize a small microorganism or to study different biochemical process in human beings, like mitosis. Or then you also, a microscope can also be used to visualize different microorganisms. And microscope, what it does is that it magnifies the object 100 times. So the microscope which you have seen in the video, they can magnify the object up to 400 times. So now the second practical which we are supposed to do in the laboratory, which is most common and you might have also heard about, is called sterilization. So sterilization is a method by which all the microorganisms, including their spores, are eliminated. Okay, so that means there is no living microorganisms present, either in the vegetative form or the spores form. Spores are the some bacteria or the other microorganisms. They will not divide, but what they do is that they can sometimes form spore when faced with hostile environmental condition, and then these spores can be used as an hibernation method. So once those harsh environmental conditions are over, these microorganisms can again start to divide from this spores. So it is very essential that in sterilization method, all the vegetative as well as the spores form of microorganisms are eliminated. So the next question which automatically comes to our mind is that why do we need sterilization? Sterilization is very important because microorganisms are capable of causing infection, are normally present in the external environment as well as on our human body. Okay, so these microorganisms are responsible for causing contamination and infection. So for doing any practical, the first and the most obvious requirement is that it should be free of microorganisms that are causing infection and contamination. So the aim of sterilization is that it should remove or eliminate all the microorganisms from the area where we are doing the experiment or from the material from which we are doing the experiment. So now, we saw that organisms can be eliminated or removed through the use of sterilization. But how do sterilization kill microorganisms? So there are several ways by which sterilization can kill microorganisms. So some of the ways are that sterilization process can actually denature protein. We all know that proteins are the functional unit of cells. So when there will be denaturation of protein, that means the proteins are not functional, then microorganisms will be killed. Then the second process by which sterilization kills microorganism is oxidation. And how does oxidation helps in killing microorganism is that oxidation creates a pore in the cell membrane, as a result of which the cell membrane becomes non-functional. And once the cell membrane or cell wall becomes non-functional, there will be no transport of materials in and out of the cell, and as a result of which the microorganisms will be killed. Then the third method by which sterilization kills microorganisms or remove or eliminate them is filtration. You all might have heard that filtered water. So filtration, what it does, they contains a membrane with small size pore. So that small size, they will not allow the microorganisms greater than size to actually pass through the filter, as a result of which the water which you are using for filtration will be free of microorganisms. Then the sterilization also helps in the interruption of DNA synthesis or DNA repair process which happens in microorganisms. So when there will be interruption of DNA synthesis or DNA repair pathway in the microorganism, this DNA synthesis and DNA repair pathway will not take place properly, as a result of which what will happen is that microorganisms will be killed. DNA synthesis and DNA repair are important physiological process which is required for survival. Then we also just now we said that protein denaturation happens due to sterilization. Sterilization also happens, what it will do is that it will interfere with protein synthesis. So there will be less protein synthesis in the microorganism, as a result of which microorganisms will be killed or eliminated. And the last method by which sterilization actually kills microorganism is through the disruption of cell membrane. So we can see that sterilization helps in the elimination or the removal of microorganisms via different pathways. So like if suppose you are asked to mention some of the ways by which sterilization kills microorganisms, so here are the ways: denaturation of protein, oxidation, filtration, interruption of DNA synthesis, interference with protein synthesis, and disruption of cell membrane. Now, there are various ways by which sterilization can be achieved, okay? And sterilization broadly can be, method of sterilization broadly can be classified into two. One is physical and one is chemical sterilization. Physical sterilization includes heat, radiation, and filtration, and chemical sterilization use chemical method to eliminate or remove the microorganism. Here are some of the name of the chemical reagents which can be used for chemical sterilization, namely alcohol, aldehyde, phenol, oxidizing agent, ethylene oxide, and other chemical reagents. So let's see briefly how some of these methods are used routinely in the lab for sterilization purpose. So one of the most widely used physical method of sterilization in the laboratory is heat method. So heat method basically consists of two types. One is wet heat method and the other one is dry heat sterilization method. So we all will see how wet heat or steam sterilization is achieved in the laboratory and how it is useful for sterilization purpose. So wet heat or steam means there we are using the steam for the sterilization purpose, and here the water is heated around 121 to 134 degrees Celsius under pressure, and this steam under high pressure actually kills all the microorganisms starting from bacterial spores to viruses. Okay, so the why actually the heated steam is so much powerful in killing microorganism or removing is that it contains very high intense heat and which is not achieved by water alone. So you can see that pressurized steam has very high latent heat, around 7 times more heat as compared to water at the same temperature. So this protocol or the heat or wet heat or steam sterilization is normally used in the laboratory for autoclaving lab experiments or lab reagents before starting any experiment. Okay, then the other way by which sterilization can be achieved is dry heat. Dry heat is nothing but just putting the experimental stuffs on fire in simple stuff, so that they can be all the microorganisms which are present on the instrument or the material which you are going to use for the experiment is sterilized. That means all the microorganisms present on the surface is either killed or removed from that place. Then the other method, which is physical method for sterilization, which is also commonly used in the laboratory, is filtration. Filtration, just now we saw heard of one example is the filtration of water. So that will remove all the microorganisms based on the size. Then the third is the use of radiation for sterilization purpose. So radiation, namely UV light, X-rays and gamma rays, are used for sterilization of lab equipments or lab reagents before starting the experiment. So UV light is commonly used, and we will see tomorrow how UV light is used for the sterilization of the area where one is going to do the experiment. Then X-ray and gamma rays are used for sterilization purpose, mostly in medicinal purposes or hospitals where the need of sterilization is very high, and they are basically used in the hospitals to sterilize, like supposedly needle is used for injection for giving the vaccine. So now that needle should not go and should not be used for another person, or supposedly by chance if that needle pricks to somebody else, so that might cause infection or contamination. So before discarding that needle or syringe to the outside environment, those needles or surgical equipments are sterilized through radiation method, basically using X-rays and gamma radiation. So the three method of sterilization we saw just now. One is heat, which is basically of two types, wet and dry. The second we saw is filtration. The third we saw is by the use of radiation method. Then there is also another method of sterilization, which is called chemical method of sterilization. And in this, basically what we do is that we use chemical method or chemical reagents for sterilization purpose. Some of the gases which are commonly used in sterilization purpose are hydrogen peroxide, nitrogen dioxide, butyraldehyde and formaldehyde solution are commonly used as a chemical reagents for sterilization purpose. Okay, so the basic requirement for any laboratory setup is sterilization. and the instrument which is used for sterilization purpose commonly in the lab is autoclave instrument. Okay, so here the autoclave uses the principle of wet heat for sterilization. So today we will see how an autoclave is used for sterilization purpose and how one need to use the autoclave for sterilization purpose, the basic principle and the parts of a autoclave. So let's watch this video so that it will be much more clearer how to use the autoclave. So I'm sharing the link which can be used to see the working principle and the parts of a microscope for an autoclave. It's a very good video, so it will be really helpful in understanding the principle of sterilization. And this autoclave method is the most widely used method in the laboratory for doing sterilization purpose.
इंडस्ट्री ऐसी है कि यहाँ पे हाय गाइस, सो वंस अगेन वेलकम बैक टू दिस चैनल। मेरा नाम है निशा और आप देख रहे हैं बेस्ट इनकम सीक्रेट्स। तो गाइस, आज की वीडियो में हम लोग एक ऐसे प्लेटफॉर्म के बारे में डिस्कस करने वाले हैं जिसपे आप लोग घर बैठे काम कर उनसे मनी अर्न कर सकते हैं। अगर आप लोग स्टूडेंट्स हैं एंड आपके पास कुछ एक्स्ट्रा टाइम है जो आप इस प्लेटफॉर्म पे आप डेडिकेट कर सकते हैं, 2 to 3 आवर्स अगर आप दे सकते हैं तो आप इवन आप लोग 500 रुपीज पर डे इस प्लेटफॉर्म से अर्न कर सकते हैं। ठीक है, अभी हम सारी चीजें डिस्कस करेंगे। इवन मैं आपको अपने मोबाइल फोन है एंड अगर आप एक इंटरनेट कनेक्शन अगर आपके पास है तो आप आसानी से आप लोग इस प्लेटफॉर्म पे काम कर सकते हैं। ठीक है, इसके साथ साथ देखो, कोई भी आपको इन्वेस्टमेंट नहीं करनी, यहाँ पर टोटली फ्री प्लेटफॉर्म है ये जिसपे आप काम कर सकते हैं। ठीक है, तो काफी अच्छी अपॉर्चुनिटी है आप सभी लोगों के लिए। ठीक है, एंड आपको किस तरीके का काम यहाँ पे करना होता है ये भी मैं आपको आगे वीडियो में बताऊंगी। ठीक है, बट गाइस उससे पहले हम लोग देख लेते हैं अपनी लास्ट वीडियो के बेस्ट शाउट आउट्स को। ठीक है, तो गाइस चलिए चलते हैं हमारी स्क्रीन पर। तो गाइस द बेस्ट शाउट आउट्स फ्रॉम लास्ट वीडियो आर
So now we saw how an autoclave is used for sterilization purpose in the laboratory. So today we studied three practicals: how to use a microscope, how to use a microscope to study mitosis from onion root tip, and the third practical which we studied was the use of autoclave for sterilization purpose in the laboratory. So let me describe each practical briefly. So we saw microscope. So microscope, the basic components which are very essential is eyepiece, objective lens, and diaphragm. And then these three combined together for better visualization of an object kept on the microscope. Okay, then we studied how does a microscope can be used to study mitosis from onion root tip. Mitosis consists of interphase, prophase, metaphase, anaphase, and telophase. So the steps starts with cut a root tip, place it on a slide, then wash it with water, just chop it into small pieces, okay, and then put 0.1 normal HCl, then put acetocarbonite stain, then cover it with cover slip, then squeeze it little bit, and then one can see put the slide on the stage of the microscope and visualize the different stages of mitosis under the microscope, namely interphase, prophase, metaphase, anaphase, telophase. We also said specifically the two important phases of mitosis are interphase, which happens actually before the actual mitosis. What is interphase? Interphase is the stage of the cell where the cell prepares for mitosis. Okay, and then cytokinesis. Cytokinesis happens after telophase and where the cell actually divides into two. So the definition of mitosis: a process by which a cell divides into two is completed after cytokinesis. Then we also saw what are the different ways of sterilization. The different stages, different ways by which one can do the sterilization are broadly classified into two: physical method of sterilization and then chemical method of sterilization. Physical method of sterilization involve heat, and heat method of sterilization is then again classified into two types: wet heat and dry heat. Wet heat means where the heating process happens in the presence of water. Dry heat where the normal heat is applied for the sterilization purpose. And wet heat is much more efficient for sterilization purpose because of the reason mentioned in the presentation. Then we also saw how filtration can help in the sterilization process. Then we saw the use of radiation for sterilization purpose, namely X-rays, UV light, and gamma rays. Then we also saw the use of chemical reagents for sterilization purpose. Okay, then we saw how an autoclave is used for the sterilization purpose in the lab. So these are the three important things. So questions will be asked basically from the presentation which will be shared with you and the videos which you are supposed to see. And for the use of microscope, the different working principle of microscope, the different parts of microscope, how microscope is used for the visualization of mitosis from an onion root tube, how an autoclave is used for the sterilization purpose. So basically the multiple MCQ type question will be broadly covering the topics which we studied today and which we are going to study tomorrow. So any doubts regarding the things which we have studied today? Anybody can please tell, or how to we can means like better the presentation, or how we can improve the presentation so that it will be easier for other people to understand. Anyway, I will be sharing the slides today along with everybody. If you get time, just watch this video once again and it will be clearer to you what we are going to study or what we are supposed to do in the microscope, use of microscope or the sterilization purpose. Anybody, any doubt? Please tell.
No, sir.
So if you watch this video, it will be much clearer, and the videos are very good and they are self-explanatory. And the protocols are also simple, like for the use of microscope, what are the different parts of microscope, which we saw in the first video. The second video was, we saw second video too. The second and the third video were basically of studying mitosis from onion root to root tip. And then the fourth video was the use of autoclave for sterilization purpose. Hopefully it will be clearer. So tomorrow again we will meet at the same time, 8:15. So please share the slide. I will share the slide with you people, so that I will send it to your emails actually. So can anybody send one email address so that I can send it and they can forward it to everybody in the lab? Not in the lab, basically in the yeah, okay. I will send this, the presentation. Okay, thanks. So I got 2 email. Okay, fine. So whomever I am sending the presentation, please share it with everybody else in the department. Okay, thank you very much for today. I will use this email ID for sending the presentation. Just 1 minute. I will use this so that I can compose. 1 minute. Online practical classes are very difficult, but anyway, we will manage somehow. But anyway, the videos which I am sending you all is very self-explanatory and it is easy to understand.